Okay, good afternoon, everybody. Pictures are allowed, and I recommend the last picture. Okay, so thank you, um, Alan and Alex, for organizing this Danilomic Symposium and giving the opportunity to present the work we are doing in Réunion Island mainly, and also in Madagascar. About my talk deals with two groups of vanilla that are poorly resolved taxonomically. What is this one? Okay. So first group is leafless vanilla from the southwest Indian Ocean region. And to give credit to the people that did most of the work, the result I will present is from Katusi Andrea Miazza, the PhD work she did under the supervision of Professor Howard Vons and Pascale Besse in Madagascar and in Réunion. And the second part of my talk will be about Vanilla planifolia and allied species that belongs to aromatic vanilla species. And this is also PhD work from Félicien Favre and tutoring by Dr. Karin Charon. Just a few words about the leafless vanilla species. This leafless vanilla I call that because the leaves are reduced to very small scales or sometimes disappeared very early in the development as an adaptation to very dry and hot conditions where they are growing in the western coast of Madagascar for the Malagasy species. These species also are credited with the medicinal properties. So they are collected by people for these medicinal properties and they are sold. What is used is a stem. So they are collecting the whole plant for the stem is used as a medicine and they are sold on the markets. So because of this recollection, wild recollection in the field, and also because of the destruction of habitats in Madagascar and the birds, most of the Aphyllus species are threatened. Aphyllae occurred in the evolution of vanilla in three instances, once in Asia, once in the Caribbean, with the Vanilla duloniana and barbellata, and also in the Indian Ocean species, where about seven species have been recorded so far. Two species are restricted to specific areas like Vanilla roscari in the Eastern Africa and here, and Vanilla phalaenopsis located only in Seychelles here. The five remaining species are present in Madagascar. One, Vanilla beauty, is also present in Comoro archipelago. So these species are puzzling for taxonomy. First of all, because they are difficult to distinguish based only on morphology. So there are two groups. One are the yellow flowers, other are white flowers. So this is very easy to make the difference. But regarding dimension of the flowers, it's not so easy. In addition, they are often found in symmetry in some regions, and there are areas like in the western here in excuse me, like in Majanga region where there are up to 4 different species cohabiting in the same territory. And lastly, up to now, genetic characterization has been done only by a plastid with a low resolution, and this analysis with plastid only can separate the white flower species from the yellow flower species. So we started with Katusia a new study recollecting large amount of big number of population all along the western coast of Madagascar, where these species are mostly found. So 23 population, totalizing 900 samples were recollected. And also two new reports of leafless species in the eastern coast, the wet coast of Madagascar also reported, and they were sampled also. And we developed integrative taxonomic approach, so trying to use several methods for characterizing the diversity and also delimitation of the species. So population genetics using microsatellite with 7 loci, follow genomics on the ITS sequences, Morphology with using 26 morphometrics variables plus color quantification. And also we collected climatic and environmental data for all the population. Going to results with morphology. So we could differentiate the different species based on color or morphology for most of the species. Sometimes morphologic differentiation was very difficult, but there are some characters that could be separated anyway. For instance, for Vanilla decariana here, which has much smaller flower than the others, and also Vanilla bosseri here that has no hair on the label, differently from the other species. From the genetic point of view, the phylogenetics analysis with ITS sequences showed seven monophyletic groups. So there was no structuration according to the country from whether the samples were coming, but there was a major clade was the separation between, again, the yellow flowers species and the white colored species. When introducing the structural analysis of microsatellite data, there were 9 groups. So 6 of these groups were congruent with the phylogenetic analysis. There was a good correspondence between the phylogeny and the genetic structure. But one clade, this big clade, were gathering 3 different species, Madagascarensis, Bosseri, and also the species from Seychelles, Vanilla phalaenopsis. It's interesting to note that there is also some signals of hybridization in these populations. So as a whole, we could decipher the taxonomy of these species using the 3 criteria we've been studying: phylogenetics, genotypes, and morphology. Morphology was only used on the Malagasy species because we were doing it with the fresh flowers, and we were sampling only in Madagascar in the last years. So with this information, so some species are identified by the 3 criteria, but some by 2 criteria, sometimes by 1. So in the 9 species, so 5 species was delimited by the 3 criteria, 5 species from Madagascar, including the 2 new species that were described from the eastern coast of Madagascar, with big flower in the wet conditions. 2 other species from Madagascar were also differentiated, and Vanilla roscari from the eastern coast of Africa was defined by monophyly and genotypes, and finally Phalaenopsis as a specific genotype. So having now quite a accurate knowledge of what are the different species, leafless species in Madagascar, we could complete the information with Vanilla valkyrie, which belongs to the same group of Aphyllus species and is present in India, but was not included in our study. And so it will be possible to maybe do some phylogeographic analysis of evolution of this group of species in the area. And also something that was interesting is that having this species differentiation, we noted by comparison with the climatic data that there was a correlation between genetic structure of the population and the climatic conditions. So there was some signal of adaptation to aridity and to dryness in this population. And we are now starting a project to try to identify in the genome of these leafless species if there are some signals that govern for adaptation to heat and dryness in this population, having probably Vanilla bosseri as a model species, because we have a gradation of different populations correlated with the gradation of climatic conditions. Also, we could got information about the status, IUCN status for this species, based on genetic diversity, also from the size of the population, the habitat, if the habitat is fragmented or not, and other sort of information like that. And 3 were classified as endangered species, 2 vulnerable species in Madagascar, and the two remaining with no special concern for them. So this was for the leafless vanilla species. Now going to aromatic vanilla species. So the emblematic species in this group is Vanilla planifolia, which is the vanilla of commerce with 2,500 tons produced yearly, more or less, with some big amount of money. Because of this Vanilla planifolia is used for agro-industry, also in perfumery and cosmetics. So it's edible vanilla. And so it is subject also to specific regulation regarding commercial and food safety for consumers. Up to now, well, before now, the only regulation was the Food and Drug Administration that was stating that only can be sold in America, in United States of America, Vanilla planifolia and Vanilla taitensis as vanilla for flavoring ingredients. Very recently, in November last year, the Codex Alimentarius, that is concerning almost all the world in the term of edible products, extended the list and defined 5 trade names associated with 5 species of vanilla: Vanilla planifolia, Vanilla taitensis, Pompona, Odorata, and Cribiana. So there is a need for these people that are doing the regulation to have good taxonomic information. And so it's our responsibility to provide the accurate information. And the relationship between all these aromatic species are not very well defined up to now. So we, starting from the collection we have in Reunion Island, collection that we have in vitro, but also under shed house. We studied using genotyping by sequencing, 137 accessions, covering 9 species and 19 interspecific hybrids. Most of these accessions were present in the shed house, so we had flowers, so we can have almost accurate identification of the species of this material. Okay, so resolution as expected was quite good. We had the 2,000 SNP. There was a good separation between Vanilla planifolia here in blue. Oh, excuse me. Here. Yeah, Vanilla planifolia in blue with the related species here, including Vanilla odorata and Vanilla pompona. Interestingly, the Vanilla planifolia was divided into 2 groups of species, in dark blue and pale blue here. Dark blue was corresponding to the clones that were propagated mostly vegetatively, so they have a higher level of heterozygosity. And the pale blue were clones that were supposed to have been quite some, for some of them we knew that they were propagated by seed. They were originating from seed quite recently, and they have a lower level of heterozygosity. As expected, the Vanilla taitensis, that was known to be a hybrid between Vanilla planifolia and Vanilla odorata, is in intermediate position in the tree. And also, we had confirmation of the separation, although it is very close to Vanilla planifolia. Vanilla sotoarenasis, it's a species that was described recently from population here in Costa Rica, on the Caribbean coast of Costa Rica, was clearly separated from Vanilla planifolia. So going back to the, because there was some debate about the validity of Vanilla sotoarenasis, so I think we can consider it's a different species from Vanilla planifolia for different reasons. First reason is a robust phylogenetic divergence from Vanilla planifolia. That was confirmed, but by totally independent work using the same strategy of genotyping by sequencing, but with a completely different set of samples. But the same species, but different sample collecting in the other places. And the structure was exactly the same. The representation is not exactly on the same point of view, but you can see here the planifolia with separated in 2 kinds of population that were named Vanilla planifolia 3 and Vanilla planifolia type 1. And here it is a group that is gathering population from that were named type 2. That were coming from Belize to north of Central America, and also the Vanilla taitensis from Costa Rica. So exactly the same structure, and vanilla taitensis that is distantly related to these two species. Another argument is the difference in size that is quite significant between vanilla planifolia and vanilla sotoarenazi, which so we probably this difference in size that is quite significant could contribute also to genetic isolation of this species, reproduction isolation because of compatibility of the pollinator. And last but not least, also there is a contrasted aromatic profile between the two species. Vanilla sotoarenazi has a high content of anisile compounds, while vanilla planifolia never has anisile compounds. And the content of vanillin, that is vanillin, and several variations of vanillin molecules, are in the lower content in sotoarenazi. So the aromatic profiles of vanilla sotoarenazi is closer to the one of vanilla taitensis than to the one of vanilla planifolia. So for all these reasons, I think it's important to separate the two species for scientific and also commercial reasons. So all the data also unable to identify the kinship of the species. Using some hybrid with a known parents, we can verify that the method was valid for identifying which are the parents of a species, if the parent is present here. For instance, this one, the citrate vanilla, which is a backcross between vanilla planifolia pompona, backcross with vanilla pompona, show as expected 25% of pompona and 75% of planifolia. This hybrid is the same as which is known here in Costa Rica as El Hibrido. And it was interesting and surprising to see that vanilla taitensis, that was supposed to be a hybrid between planifolia and odorata, indeed the female parent of vanilla taitensis is closer to vanilla sotoarenazi plants than to vanilla planifolia. So this gives some new insight about the history of the introduction of vanilla taitensis in French Polynesia, because now this species is especially cultivated in the Pacific. Historic records say that it was introduced by Admiral Ferdinand Hamelin from South America, going through Philippines, but apparently it's not true. He sailed directly from Central America to French Polynesia, and giving the area of extension of vanilla sotoarenazi, maybe because no one has seen one vanilla taitensis already still in Central or South America. So if someone wants to find some remaining vanilla sotoarenazi in vanilla taitensis in Central America, maybe the best place to search for would be in Central America from Choco to Belize. So it's quite a good place for exploration. Okay, and thank you for your attention. And I promise you the last slide, if I have a few seconds, just to, if I can do some advertisement here. So we will be organizing Vanilla Congress next June. So there is still some place for registration. And so if you are as in love with vanilla as I am, so you are very welcome to join us in June. Thank you very much.
Thank you very much. And we have time for 4 or 5 questions. So plenty of time. Do we have any?
Thank you very much for your great talk. Just out of curiosity, was it challenging to sample leafless vanilla?
If it was what?
Was challenging to sample for...
I have difficulty. That's quite difficult to extract DNA from a stem because you have to collect stem. So it's not so difficult. What is difficult is to travel in Madagascar. That's difficult. But then to collect the sample is not so difficult. And for extraction of DNA, it's not so easy, but we managed to do it.
Thank you.
Thank you very much. Anyone else? I see. I have a question, if I may. I will use the microphone since. Just a curiosity, there are two species that you showed pictures, Humboldti and Pereri. Do you know what visits these flowers? The yellow one with, the
It's a bees. We have studies. For which one we were talking?
Humboldti, the yellow one with the
Humboldt.
marks in the labellum.
Umbloti is visited by bees. It's what we've seen in Comoros. But there are a big population of the species in Comoro, and I don't remember the name of the bee, but the bee was identified, including
So it was
with pollen.
Interesting. Thank you very much. Anyone else? Oh.
Thank you for the presentation. I would like to know if you have data on chromosome information of this species, if there are polyploids or among the hybrids.
Which species are we talking about? Polyploid is
the complexes that you
in the leafless vanilla, we have a little information about the genome and the number of chromosomes. Probably recently we did a count, it was 32 chromosomes, the same as vanilla planifolia. The size of the genome seems to be bigger than vanilla planifolia, maybe the double of vanilla planifolia, like something like 210 gigabase. And for the ploidy, we don't know.
The hybrids, no information.
Which hybrid?
Well, the three hybrids that you showed in the planifolia complex.
The hybrids. No, that is most of the hybrid we don't know the number of chromosomes. In Titaensis, for example, which is an hybrid, there are diploid plants and there are also some tetraploids varieties.
Nice. Thank you.
Thank you very much. Do we have any more questions? I don't see. Okay, so thank you very much.
Thank you again.